Okay, great. So thanks to all of the attendees, particularly the virtual attendees who have stuck it out through the morning. I see there's still quite a large number in the chat, and I realize I'm the last barrier between you all and lunch, so I will be on time, and I want to thank you for sticking around. I'm Jason. I'm the sales manager for IDEA Biomedical, and we are represented by Isogen, so thank you very much for the invitation to present our technology here. Today, I would like to take the opportunity first to introduce IDEA Biomedical and the type of automated microscopy that we offer to the market. Then I'll offer you a lot of pretty pictures showing the types of images that we can obtain on our microscope, and in general, the type of images that can be obtained from different types of microscopy. I think that Professor Debin's presentation before showed very nice ones of that sort, as well as for the analysis. So I will show the type of analysis that we can do on 3D cell culture within our platform, and hopefully give you some ideas for what you can do with your own work. So with that, I'd like to begin by describing our company. So IDEA Biomedical is a small joint venture between a larger company called IDEA Machine Development, Design, and Production. So that company has over 2.5 decades of experience producing and designing electro-optics and precision motion machines specifically for the silicon wafer industry and the industrial printing industry. They're strategic suppliers for some of the largest companies in the world, making microscopes that go into clean rooms and can never come out. And so they joined forces together with biologists from the Weizmann Institute of Science several years ago to bring their microscope and computer vision technology to the life science realm. And that gave rise to who we are at IDEA Biomedical. So we offer basically a workflow solution, as was pointed out in Professor Debin's nice presentation before, the whole workflow for automating the work with biological samples in multi-well plates. So what we take in is a multi-well plate, such as a 96 well plate, round bottom, flat bottom, whichever you'd like. We are now offering a stacker unit that can interface with a robotic arm that will pick up a plate, plop it into our Hermes microscope, which is our flagship product. It is a wide field automated microscope. The images that come out are then analyzed and give the output data for the users to use in downstream data mining or other types of analysis. So in this fashion, from a single supplier, you can get a fully automated workflow for your 3D cell culture needs. One unique aspect of the Hermes I would like to point out is that we move the objective in all 3 dimensions, X, Y, and Z, and leave the sample stationary. This makes it much gentler on the sample because you don't have to agitate it or jostle it about while you're moving from one field of view to another. And it also enables the imaging to occur much faster. We're compatible with all types of microscope objectives, a range ranging from 2x up to 60x with air and up to 100x with oil or water. And this covers indeed the 20x magnification that Irene mentioned for her SPA chips. It's very easy to exchange the objectives in our microscope. As you can see in the movie, you just plop it in, and it's ready to go. Its custom holder will align it with high precision. And here you can see how the illumination will move as the objective is moving and leave the sample stationary so that we don't disturb your gentle and precious 3D cell culture. The software too controls exchange of the objectives so that you can have multiple magnifications within the same microscope and make it ready to go for any number of experiments. Anything from low magnification for general overview to high magnification for highly detailed intracellular studies. We offer it with a combination of either 4 or 7 colors, always including bright field. It's compatible with on-stage live cell conditions. We have very fast acquisition, having either 1 camera or 4 camera configurations that are able to image a full 96 well plate with a single field of view in 4 colors in about 100 seconds. We also offer video for fast dynamic processes up to 40 frames per second. The autofocus is hardware-based using laser reflection, and that is compatible with all objectives and all sample types. Speaking of that, we accept anything that's an SBS sample standard or microscope slide, dish, EVD well plate, whatever you'd like. And as I mentioned before, a broad range of objectives. And I'd like to point out that we offer water and oil immersion objectives and fully automated dispensing of the immersion media for each of those. If you'd like more, I'll make a pitch to our sponsor, Isogen, so please reach out to them in your respective territories. So let's get on to the pretty pictures. So basically, any type of workflow involving microscopy will have a number of steps. The first one is image acquisition, where, as you may know, if you've performed this yourself, you want to do a variety of different tasks. This can include imaging throughout the sample volume through a Z-stack, obtain a time lapse, so images over a time course to see the development of a single sample, or a video in the case that you want to see fast dynamics. Once you have the images, you may want to process them to change how they're structured or how they appear. This can include fluorescence deconvolution to remove out of focus light, slice projections so that you take all the fluorescence over a volume and project it onto a single image, or select a single best representative slice from a Z-stack or a time series. Once you have the images you want, you analyze them through the process of segmentation. Segmentation is the way that a computer will identify which pixels in an image represent the object of interest, and then use the combination of those pixels together in order to extract morphological information about your samples, as well as any type of fluorescence intensity that you're interested in. And that is the output data that is most commonly used in order to extract images from a sample using microscopy. If you're going to want to do this with automation, the reasons that you would do this is to obtain the benefits of speed, because the robots are able to do it much faster than we are. And by doing it with an automatic robot as ours, you can also free up your time so that you can go do something else and work and gather data even when you're outside of the lab. And ideally, you don't want to do much programming. You want to focus on biology. That's your job. So we endeavor to make it as easy to use as possible. So here's some examples of a Z-stack through a relatively large spheroid, going over 200 microns with 50 micron step sizes. So as I mentioned before, once we have the images, some of the processing steps that we may want to do would be best slice selection, such as we can see here. You may also want to do a quick overview before you do that. You want to see all of the spheroids or organoids in a multi-well plate so that you can identify particular wells that may not have formed properly. These are particular spheroids in a ULA 96 well plate. And so you may want to identify those that have malformed. Here we can see easily identify different sizes, different shapes through visual inspection. We also may want to visualize it in terms of the fluorescence to see if we have a difference in cell death, as we can see in the red channel here, or the presence of expression of proteins as in the green channel. As well, we can do a Z-stack in the fluorescence channels, and then we can take these and perform operations such as fluorescence deconvolution in order to remove the out-of-focus light that's coming from above and below the sample plane in the wide-field imaging modality. And here we can see the effect of the deconvolution on the best slice, where it's easy to appreciate the enhancement in contrast and the reduction in background fluorescence, in this case without compromising on speed. So once we have our images in hand, we will want to perform analysis. So I will first introduce that with our Athena software, and then show a few examples of that. So within our platform, and if you've also done it with other platforms, it may be similar, where you have segmentation masks that are presented on top of an image, as we can see here on the right. And through adjustment of a number of parameters, you can, for example, the threshold, as is shown here, you can identify larger or smaller portions of the samples of interest based upon the characteristics that you visually inspect during the analysis parameter calibration. And once you have the parameters defined, it becomes repeatable. So even if it's not exact, you will always be performing the same action, which is something that human and manual analysis is not able to achieve. Once the data is extracted from each of the images, the visualizations that can be helpful include heat maps, so that we will take the quantified metrics and represent them in a colorimetric span, and then place them into a representative plot showing the outline of the sample type, for example, a 96 well plate. That data can then also be aggregated together, together with concentrations to form dose curves, as we were seen before. For time-lapse, we can also see time evolution plots, and 2D scatter plots of different metrics are able to identify potential populations of interest. So within Athena, we offer a broad range of applications that are ready to go out of the box and all included in the software package. Most of them are cell-based, given the history of high-content screening, but we do have specific applications relevant for the identification and measurement of spheroids and organoids. So first, a couple of examples with spheroids where we have a little bit more experience. So this includes identifying multiple objects or spheroids in a large multi-well plate, in a large dish of a multi-well plate, the dose curve calculations, also quantifying the quality of shapes that a spheroid has formed, live dead assay, and cellular co-culture. So in this example, we can see a number of spheroids that have been formed in one of the wells of a 6-well plate, and we can see them dispersed at different regions of the plate, and outlined in orange, I hope it appears on your screen, are the spheroids themselves, along with the unfortunate debris that also comes along with the cell culture. It's notably difficult to extract the signal from the noise in terms of the debris. However, using a number of the quantified metrics, we're able to reliably identify which of the objects in here are spheroids based on their shape, and exclude the ones that are going to be debris and should be ignored for any type of analysis. So this type of example is useful for counting, for example, just simply how many spheroids are there and getting an idea for the distribution of their sizes. Again, we have a plate preview where we have 2 compounds that have been applied to these tumor spheroids. We can see in rows 2, 3, and 4, so B, C, and D, that as we move to the right, the spheroids appear to get smaller, particularly in this region, whereas in the bottom, E, F, and G, we see that there is a compound that's been applied but no treatment effect. Again, we have a malformed spheroid that we ignore for our analysis. So here's the heat map, the same as was shown previously, but now with the information accompanying it, where we can see visually through the colorimetric representation of the spheroid area that compound 1 leads to a decrease in the size of the spheroid as the concentration is increased, whereas compound 2 does not. In this way, we're able to get a quick estimation for the EC50 value for the compound in question. If we want to look at the quality of spheroid or organoid formation, we can do that as well through quantitative multi-parametric categorization. So within our software, similar to some of the others, we have a number of metrics that are extracted, such as solidity, the axial ratio, and fitting the object with a polygon called a convex hull. And with that, we're able to identify spheroids that we want and ignore the ones that we want that are misshapen or malformed. We can do this across a multi-well plate. This particular multi-well plate is a ULA round bottom 384 well plate. We imaged this one at 4x magnification in bright field, and we were able to take advantage of a unique pre-mapping focus feature that we have on the Hermes to image the entire plate in 1 minute and 32 seconds, so about a quarter of a second per well. During analysis, I used the aforementioned multiparametric categorization to identify the wells that have properly formed spheroids and which wells have spheroids that should be ignored. Having this characterization makes it very easy in order to identify the area of the spheroids that we want to be studying, to count how many there are, and also look at scatter plots to see how they compare to the noise or other types of spheroids that we're ignoring in this particular sample. This, for example, could be a preliminary step in performing a live-dead assay. So here's some examples of, here's an example of a live-dead assay in a spheroid that we did. So we have the nuclei labeled in Hurst. We have the live cells labeled with, I've forgotten what it is, a green dye, calcine AM, and the dead cells labeled with PI in red. If we do simply just a cell count, we can identify how many cells are identified in blue, green, and red. This particular sample was designed with an increasing seeding density from top to bottom that becomes visually apparent through the colorimetric heat map that we can see here, with fewer cells identified in all three color channels, blue, green, and red, at the top, and more cells being identified at the bottom. Then, for the live-dead ratios, we can simply we can divide the cell count in each color by one another. So in the top, we take the number of green cells divided by the blue cells, and this gives us the live ratio. We can then take the number of dead divided by the total number of nuclei, and that will give us the dead ratio. And the ratio of green to red will give us the live-dead ratio. This plate was also prepared with an anti-tumor treatment compound on the right half of the well, and we can see particularly that in the red channel, with the dead ratio, we have a much higher number of dead cells on the right half of the plate as compared to the left, and that the number of dead cells relative to the number of live cells is higher in these smaller spheroids. I think this was a comment that Professor Debon alluded to previously, and this is one of the methods to identify it in single spheroids without having to use his bulk method ATPS assay. In terms of cellular co-culture, this is an example that we got from a client who had cultured a type of native tissue-specific healthy cells in red, and the tumorigenic version of those same cells in green. They were looking to identify the metastasis or movement of the green cells into the red. So then in this particular example, we simply identified the area in green and the area in red. Again, there was a cell seeding density variation from left to right that we can see represented colorimetrically by the area of the spheroids, and then taking the ratio of those two with increasing treatment conditions gives rise to this 2D metrics, which is not intuitively not necessarily intuitive in terms of the readout, but does show the power of this type of analysis to get multiple data points in parallel. So to wrap up, I would like to show a little bit of the experience that we've had with organoids, identifying protein expression zones, and looking at the detection of beating or contraction in cardiac organoids. So here's an example of a cardiac organoid that was labeled with 4 different colors. So we have all nuclei in blue. Again, this is a wide field imaging microscope, which you may have had experience with in the past. This is EDU, so this is the replicating nuclei within the organoid. And these are 2 proteins of interest that they're looking to associate with nuclear DNA replication. So within each color channel, we can identify the organoid itself. We can identify and count EDU positive replicating nuclei, and we can identify the protein positive regions of the organoid. We can see, for example, on the right-hand side, the 2 proteins of interest are absent but heavily present on the left. So we can then count and classify each of the nuclei in a similar type of quantitative classification as I described before. So we identify all of the replicating EDU positive nuclei in white. The ones that are present only in the protein 1 channel, we outline in orange, only in protein 2 channel, outlined in pink, and both the triple positive cells are outlined in yellow. So in this way, we're able to readily extract 3 different populations from a single experiment. Then we can take the ratio of the double and triple positives compared to all of the EDU positive cells. So this is the ratio of replicating cells within the protein regions of interest relative to all replicating cells. And in treating with different compounds that they remained confidential to me, but they told me compound 1, 2, and 3. We can see the first compound shows a nice dose response where we see an effect of reduced replication inside these protein areas as compared to the other 2 that appeared to have mixed or no results. And overall, in this case, we are able to analyze nearly over 31,000 cells in about 2 minutes of analysis. The image acquisition took a little bit longer, about 10 or 15 minutes, and the analysis about 2 minutes. And finally, this was a cardiac organoid that was undergoing contraction that for me as a trained physicist was quite exciting to see. So this is a single image from 12 different wells that they had prepared for us. Sorry, 10 different wells that they had prepared for us. And we had 2 different time points looking at the same organoid at 2 different time points, but first under control conditions and later under treated conditions, where they added a compound that, again, they didn't tell me what it was, but we can see that in the same length of the video, being 15 seconds, that the contraction of the organoid under the control conditions showed 4 contractions, so about 16 beats per minute, whereas it went up to about 20 beats per minute under treatment conditions. And this was all imaged in a fully autonomous fashion by the Hermes, and a repeatable analysis through the Athena software platform. So, in summary, the Hermes that we offer is a broadly applicable platform, not only for 3D cell culture, but everything from tissues down to individual bacteria, and for a number of assays, if you want to do protein expression, fluorescence in situ hybridization, specifically for RNA is one of the examples, nuclear translocation, and many more objects. So with that, thank you very much for your attention. And if you have any questions, please ask now if you're interested. And if you're interested in more of the product, please reach out to Alike or Nurdia, who are from Isogen. I have this improperly labeled. So thank you very much.
Thank you very much, JC. Do we have any questions? There's a question. Yeah.
Thank you for your presentations. I'm wondering what this device, or is the range of the imaging plate that is applicable? For example, can it read like EBD plate, or it needs to be kind of like a certain brand?
Yes, it can be. EBD plates are fine. So EBD have very nice, high-quality products. Particularly, their 96 well plates are very nice. I'm not sure if they're low ULA, but they also have a number of slides, multi-chamber slides. So 8 chamber slides, 18 chamber slides, and those will fit into the Hermes through an adapter that we offer.
Yeah, thank you.
There's also one question in the chat. What set range can the Hermes scan, and how deep can it go? So the Hermes is able to image over a very deep range, several hundred micrometers without a problem. And in fact, we regularly image zebrafish, which are about 1 millimeter thick. So we've never had a problem going deep enough. I will mention it is a wide field microscope, so it depends on the question that you want. But certainly, in order to get a good characterization of the morphology of a spheroid overall, the Z range that we are able to achieve is useful for the ULA plates, where there's a bit of variability in the best focus plane. So we haven't had any trouble
in terms of the Z range that we want to use. But again, it will depend on the sample type and the objective magnification that you want to use.
Okay, and there's a last one. Well, can the autofocus work with round bottom and U-shaped plates?
Yeah. So we have defined a specific patented method in order to image round bottom plates, because since we use the reflection of a laser beam, we had to devise a special method in order to do that, and they're no problem. Any of the magnifications that we support can be used with round bottom plates. However, as I mentioned before, sometimes the round bottom plates are a bit thick, and so high numerical aperture or high magnification objectives may not go through the sample just because of the working distance of the objective. But the autofocus is not a problem at all for round bottom.
There's a question now here in the office. Martin?
Hi, Jason.
Hi.
You said that about 30,000 cells were basically analyzed in, what did you say, two minutes?
Yeah.
How long did it take? Because that's the analysis time that you have when you have everything set. But how long did it take to optimize the protocol to bring it down to 2 minutes?
So for me, it took maybe 10 minutes. I have quite a lot of experience in defining the analysis parameters. What I find is when I'm training new users or during a demo, it takes about half a day for somebody to learn the software and identify their own parameters. That obviously decreases with experience, but I think rather it is a one-time time investment, that if you make the proper plate and the proper first experiment, that you know the outcome that you want, you can identify parameters, within a relatively short period of time, we'll say 1 hour, that you can then use and repeatedly apply to any number of plates that you want in the future. So it's a one-time time investment for future replicability and with any user.
Thanks.
Mm-hmm.
And well, there's a last question about prices. Could you give some idea about the price of the basic microscope analysis software? Yeah.
I will let you reach out to Alicia and Nuria, and I think that we could, the best idea is to learn a bit what you need and make sure it's going to be the right fit for you. Alicia and Nuria can help do that, and then we can organize a call together.
Thank you. And thank you, Jason. Actually, there's the last presentation now, so you were not the last one before lunch. But,
okay, schedule changed. I'm off the hook. Thank you very much.
Thank you. I saw that Adelina is prepared.